Welcome to third year BSc Botany. I am Amisha Shirodkar-Pednikar, Assistant Professor in the Department of Botany at Parvatibai Chogle College of Arts and Science, Autonomous, Marga. Today we will be seeing the course Cytogenetics and Plant Breeding. The unit is Quantitative Inheritance. The module name is Monogenic versus Polygenic Inheritance. Outline: we will be seeing monogenic inheritance and polygenic inheritance. Learning outcomes of this module will be: understands the concepts of monogenic and polygenic inheritance, differentiates between monogenic and polygenic inheritance. Introduction. Let us first see what are monogenic and polygenic traits. Monogenic trait is a characteristic that is produced by a single gene or a single allele. An example of monogenic trait is the ability to roll your tongue, or in plants, an example would be the round or the wrinkled seeds of pea plant. Whereas a polygenic trait is a characteristic that is controlled by two or more genes that are located at different areas on different chromosomes. Examples of polygenic inheritance would be human skin color, human eye color, the height, weight, intelligence of people, and amongst plants, one example would be the kernel color of wheat. So the key difference between monogenic and polygenic inheritance lies in the number of genes involved in the determination of a particular character. So if we have to define polygenes, it would be two or more different pairs of alleles with a presumed cumulative effect that governs such quantitative traits like size, pigmentation, intelligence, etc. These contributing to the traits, the alleles which contribute to the trait, are termed as contributing alleles. They may also be referred to as effective alleles, and those appearing not to do so are referred to as the non-contributing alleles, may also be referred to as the non-effective alleles. So, in monogenic inheritance, one trait is determined by a single gene, and in polygenic inheritance, one trait is determined by two or more genes. So thus we can say that in monogenic inheritance, it refers to the kind of inheritance whereby a trait is determined by the expression of a single gene or a single allele and not by several genes as seen in polygenic inheritance. Having understood monogenic and polygenic traits, let us try to understand what is qualitative and quantitative genetics. In order to understand qualitative and quantitative genetics, let's see what are qualitative traits. The classical Mendelian traits are the ones which are qualitative in nature. Now, these traits which are qualitative in nature are easily classified into distinct phenotypic classes. So, there are these distinct phenotypic classes which are under the control of only one or very few genes, and they have no environmental modifications to the gene effects. The example here would be the height of the pea plants, either tall or dwarf, or the seed type of pea plants, either round or wrinkled. So these qualitative traits are usually determined by a single gene, that is, they are said to be under the control of a major gene, and therefore these qualitative traits are the monogenic traits. As against that, let us see quantitative traits. Now, these quantitative traits are the ones which have a spectrum of phenotypes. These traits exhibit continuous variation that can usually be quantified, and it can be quantified by measuring, by weighing, by counting, and so on. Traits such as the grain yield per acre, or the human skin color, or the kernel color in wheat are all quantitative traits. These quantitative traits were earlier referred to as the metric traits, and these have continuous variability. So these quantitative traits or the metric traits, they tend to be more complex. and they are usually controlled by multiple genes. These traits, like I said, form a spectrum of phenotypes which blend from one type to another, and thus they cause continuous variation. So these quantitative traits are the polygenic traits. So we can now finally say that in polygenic traits, there is each contributing non-allelic gene, and it has a small and relatively equal effect. The effect of every allele is additive. In polygenic traits, there is no dominance, no recessive characters, there is no epistasis or no interactions which are observed. And the value of the trait is influenced by the environmental factors. Now, in summary, if we have to differentiate between the monogenic and the polygenic traits, the monogenic traits, like we saw, are the classical Mendelian traits. The height of the pea plant, the seed type of the pea plant. So these are the classical Mendelian traits. Whereas the polygenic traits are the non-classical Mendelian traits. In monogenic traits, the phenotypic classes are under the genetic control or the genetic influence of only one or very few genes. Whereas in polygenic traits, they are the quantitative traits or the metric traits, and they tend to be more complex. These are usually controlled by multiple genes. In monogenic traits, the phenotypic classes are very distinct, whereas in polygenic traits, the phenotypic classes have a spectrum. The spectrum of phenotypes which blend from one type to another to cause continuous variations. In monogenic traits, the traits may be dominant, recessive. No such dominance or recessiveness is observed in the polygenic traits. In monogenic traits, they may express epistasis, they may express allelic interactions. However, in polygenic traits, no such epistasis or interactions amongst the different loci contributing to the value of the trait is seen. Also, in monogenic traits, the traits are not influenced by environmental factors, whereas in polygenic traits, the value of the trait is influenced by the environmental factors. Here, students, are my references. Thank you.